Can you also?
So yeah, I am in Stockholm now, and my group is based at KTH, which is the technical school here, and we are also part of what is called Sci Life Lab, which is a joint activity of 3 universities in Stockholm area. And this is my group. And today, what I will talk to you about is mostly the work done with Movitz, who is an information theorist expert, and he came to us to study neuroscience with his expertise in in information theory. And obviously, other collaborator, Mikael Skoglund and Pavel Herman. Pavel is a neuroscience person, and Mikael is another communications information theory person. So yeah, our work started with this idea of tuning curves. And if we quickly survey tuning curves, this is the work that started long, long ago with Steve Kuffler and then Hubel Wiesel and so on. And ever since we have been measuring tuning curves in different sensory areas, but also other cognitive areas. But if we stick to just early vision or early sensation, then we have largely 2 classes of tuning curves. You could say monotonic tuning curves. For instance, here, this would be an example for olfaction. So some odorant is presented, and you have the sigmoid curve. Similar thing could be there for sound, for instance. So you can get these sigmoid-like tuning curves. But more interesting, perhaps, and more familiar we are with are the non-monotonic tuning curves. For instance, the classic example of single peak tuning curves that we see in early visual areas. Also, if we go further away from just pure sensation, for instance, place cells, they would be an example of a single peak tuning. Then 2005, we have this discovery, quite an amazing work from Mosers, where they described for the first time, I think, the periodic tuning of cells, that neurons are not just tuned to a place, but they actually are tuned to many places, and they are more or less periodically distributed. Now, this is the case for stimuli where we can nicely arrange them on some axis. But in many cases we can't, and odorants are perhaps one example of that thing. And there what we would then say is that, well, maybe the tuning curves are not periodic, they are definitely non-monotonic, but also aperiodic. And I think this is the spectrum of tuning that we observe in the brain. And what we started to ask was, like, why such diversity of tuning curves in the brain, in different brain areas for different modalities? I mean, one answer to this could be that, well, it's biology, whatever works, works, and that's it. But maybe there are certain advantages of one particular kind of tuning over other, and that's what we were interested in. So this is how we formulated then the problem. This is like a typical engineer's view of looking at things, that you have some stimulus, it gets represented in the brain, and obviously there'll be some error contributed by the sensors. Then there will be tuning of the neurons, and once the neuron has received all the input which is specific to its tuning, it will also add its own noise, and eventually we will have some output. And what we want is to decode what was the input. Now, many people will disagree with it, that this is the actual goal of vision or any brain process, that, I mean, why bother about just representing the stimulus? But I would like to argue that this is just a way to formulate the problem. This X has to be mapped to something. It could be Y. Just mapping between X and X hat makes it just more tractable. And we have then these nonlinear functions which map these stimuli that come into a neuron to the output. And this is how we can then look at it, not seeing it as single neuron, but multiple neurons. And typically we are used to looking at it as a pair. So these are 2 tuning curves, and what they do is that they carve out this encoding manifold, if you wish, which is living in n-dimensional space. And the dimensionality of the manifold is, of course, the dimensionality of the stimulus space. Then. So in this case, if it's just an angle, then it's a one-dimensional curve in n dimension, n being the number of neurons that we have recorded. And the way we would always see it is that there is always noise. As I said, that there are at least 2 sources of noise we can characterize observation, which is the sensor noise and the channel noise, which comes from the the neurons. And because of that, what we will observe is some kind of noisy response for a given stimulus. And of course, the challenge would be then to use these pink dots to infer something about the stimulus. And we have a very nice machinery for this. We typically use Fisher information to calculate how good can we be given a certain noise, what will be the estimation error, and we can analytically calculate it or numerically calculate it. Once we know the shapes of these tuning curves, we just need the derivatives and the covariance matrix. And that gives us the bound on how good we can be in interpreting it. And there's a lot of work in this in terms of like, here I'm assuming pretty much uncorrelated noise, but this noise could be correlated. And there's this very nice review long ago where Peter Latham, Alex Puget, they talked about how different kind of correlations may be helpful in decoding the stimulus. So for instance, in some cases, it may be useful that the neurons, they are anti-correlated, and therefore you can find a linear separability between them, as opposed to a positively correlated case where there will be some ambiguity. And in fact, in such a scenario, it would be better that nothing is correlated. But a more general picture emerged slightly later, where if you now think of this as the encoding manifold, actually, when we think of noise and the correlations in the activity of neurons, what we are looking for is that this product, the correlations, they should be pretty much orthogonal to this encoding surface. So this would be the encoding surface, and these arrows, they mark the information direction. And what you want to do is your noise should be such that it is orthogonal to this encoding surface, and that's what gives you the best performance. Now, this is a view that has developed where we say that, okay, take this tuning curves a priori that they exist, and then comment about the role of noise. And what we did here is that maybe we reverse the question, and we could say that given the noise exists, what would be an optimal shape of a tuning curve? Could we reverse this question basically? Instead of, given tuning curve, what kind of noise is useful, what kind of correlations are useful, we reverse it saying that given the noise, what kind of tuning is useful? The formulation is very simple. So as I said, that there are two places where noise enters in this system. One could be the sensor noise, which we call as observation uncertainty, and then channel noise, which I think arises in the neuron membrane, which could be correlated, could be independent. Now, given these noise, let's say this is the encoding surface, you have some true stimulus, but both these noise, they will map in any given trial, they will map your activity to some place close to this encoding manifold, hopefully. And then the task of the decoder to estimate X hat would be to find a place where you put this projection onto the encoding manifold. And the simplest thing we can do is that, project it to the closest point onto the encoding manifold. And that gives you f of X hat. And from there, you infer your estimated location. Now here, in this case, the error seems to be small, so you get mapped pretty close to the original stimulus, and this is what engineers would call as a weak distortion. But there could be other scenarios where this distortion could be quite large, or you could be in some region of the encoding volume where the noise is such that even though you are present at this stimulus, but the closest point where you can map to is far away from the original stimulus. So this would be pretty catastrophic. Like you are seeing I guess it's US, so I have to say baseball. So you're looking at a baseball which is moving, and you're looking at the seam of it. Maybe it's wobbling, but you could make a very catastrophic error and gets struck out. So this is what they will call large errors, and large errors meaning catastrophic in some sense. And obviously it depends on the signal to noise ratio, that depending on whether you have a high signal to noise ratio or low, you will be in a threshold distortion regime or weak distortion regime. But it also depends on where you are on the encoding space. So first thing that we started was to really think about these different noises, whether they are any different from each other or they are the same, because I think as an experimenter, we perhaps will never see observation uncertainty and channel noise separately. or maybe we can. There are some signatures, perhaps. So this is how we then formulate it, exactly what we did. And for weak noise, of course, we can do it analytically, and that's what is very satisfying. So the trick is very simple. So it's yeah, very simple calculus that you take the Taylor expansion around the location where your stimulus is, and then get the covariance of the activity. And from the covariance, then we can estimate the error, how much error will there be. But what was interesting in this simple calculation is that even though we add the noise as additive, so this noise here coming from the sensor side, this is additive, but it turns out that in the activity, it gets multiplied. So here we have the sigma of n, and now there's a multiplication here. So that means that even though this noise is added, on the activity side, you will see it as a multiplication. And multiplication meaning that this observation uncertainty will also have some tuning, because of course it will vary according to the stimulus. And yeah, so noise correlations that we observe should be stimulus dependent. And this is something that we already know from literature. So this is an old work from Gustavo Deco. They analyzed the activity of neurons when different kind of stimuli were presented. So of course they have a tuning in terms of the normalized activity depending on what stimulus direction you are presenting. Each row is a different kind of stimulus, but still there is an orientation sense here. So therefore we can look at stimulus direction. What was interesting in this data was that there is a tuning in terms of trial by trial variability. So basically noise correlations that we were looking at. So in some sense, one could argue that this one source of tuning of the trial by trial variability could be actually the sensor noise. Maybe, maybe not. But if we further solve this for weak noise, we come to a point where we can actually analytically get a sense of like how much error will there be only in this weak regime. And there, again, this noise appears as a multiplication, and the channel noise appears as an addition. So there are some interesting differences whether the noise is added before it gets mapped onto the tuning curves or after that. So the key differences between them were that observation uncertainty, it doesn't depend on the pairwise product of the tuning curve gradients. So that's here. So this part is from observation noise. And here we have the gradient of tuning curves, which is squared. So therefore the sign doesn't matter, but it matters for channel noise. And the error is independent of firing rates. Channel noise depends on the mean of the firing rates. At least for this, narrow tuning does not guarantee a reduction in noise. Whereas if it were the channel noise there, if the tuning becomes narrower, we get a better coding. So this is something that one could now develop and perhaps try to measure observation uncertainty and channel noise. There is a great similarity between this observation uncertainty and what is called as information limiting correlations as well. So, but we are still not very sure like how these two ideas differ from each other. We have a feeling that they are two different concepts, but yeah, that's something we are currently looking into. But this notion then of weak distortion and threshold distortion, that actually was interesting. And we said that, oh, could we use now this to say something about the shape of the encoding manifold? And obviously, if you want to now think about it in the following way that, well, this error happens because you have some noise and you get mapped here. But imagine this curve was stretched quite a lot. That means that these neighboring points, they were close by. So in that case, the stretching of this tuning encoding manifold will actually try to reduce this error, the weak distortion. Now, if you keep stretching them, because neurons have a finite firing rate, you will at some point run out of spikes. So meaning you can only represent a small range of stimuli. So a solution to that would be, well, fold it because there's enough space in this 2D. And this, by the way, applies in ND as well. So you can fold it. However, as soon as you start to fold your tuning curve, you run the risk of threshold distortion. So that means to avoid threshold distortion, what you want is that if you have folded the tuning curve, you want them to be as far as apart. But to increase the range of stimulus variable that you can represent and to reduce weak distortion, you want it to be as stretched as possible. So that gives a constraint that tuning curves should be somehow locally curved and globally flat. This idea that tuning curves get folded, I mean, here we just make this argument from the point of view of like, okay, I want to represent large numbers with high resolution. And this argument, by the way, I think first perhaps made by Lafayette as well. And now there is a paper from bats where Nakum's lab in Israel, they let these bats run over long distances. Unlike rats who run over short distances, they can fly over 6 meters and 200 meters and so on. So they have this amazing setup. So here is an example of the same bat running in 6 meter, and these are, I think, the same neurons as well. So first they look into 6 meters and they find that tuning curves are like classic place fields. But if you let the animals run over longer arena, longer distances, then you suddenly start to see that the so-called place cells, they actually develop these multiple fields. So in a way, it looks like that for small range of stimuli, when the animals have to represent small distances, they actually rely on single peak, which will be something like this kind of manifold. And as soon as they enter in a region where they have to represent large values, their encoding manifold gets folded. And here I'm just drawing a schematic of an Archimedes spiral, which is just an optimal way of packing things. So with this, by the way, this is not something new, not that we came up with or Nakum or Ila, but originally this idea is due to Shannon, like many, many other ideas. So he has this figure in this classic paper where he says that how you can put a line in a square, and basically the idea is the same, that you are stretching the encoding and folding it so that you can have more capacity. But this gives us now some feeling for like how to think of the encoding manifold shapes and this kind of errors. So we have multiple peak tuning curves, which will create some kind of spirals. Single peaks will create individual loops. And these monotonic one, whether this is linear or sigmoid, they are going to create a manifold which is going to be just a line. And now we can automatically start to think like what will happen, that monotonic tuning curves, they pretty much guarantee you that there is no threshold distortion. On the other hand, when you have multi-peak tuning curves, you always run this risk of threshold distortion. However, here you have a very high resolution, meaning you can in principle represent very large values. Here you possibly can't. And the single peak tuning curves are somewhere nice mix of this too. So based on all this, we came up with these heuristics that on one extreme, we have these folded tuning curves or encoding manifolds and completely stretched out. So in this, what happens is that these manifolds are good with low channel noise, low observation uncertainty, obviously because there's a risk of threshold distortion, and what you want is as small error as possible. And most likely you also want very weak background activity. On the other hand, if you have correlations, then these are also pretty bad with if the sensor noise is correlated. And by contrast, these are very good. Other thing, we hypothesize that such manifolds are good for low dimensional stimuli as opposed to these ones. And here you need many, many more numbers. The reason we think about such stimulus dimensionality is that what we are thinking now is well, there is this space, but what you can imagine is that this tuning curve will be modulated if these cells were also, let's say, contrast sensitive or color sensitive, meaning this available space will be filled by that. Now, here you can do that because there is space, but if you have this kind of spiral where you have folded things, there's very little space left, meaning if you have high dimensional or so-called conjunctive cells, It will be very difficult to pack things. So therefore, we think that maybe this is low dimensional, and here you can have much higher dimension. So from here, we had some notion that take this idea of noise, whether or the decoding, that whether you can afford weak distortion or strong distortion or large distortions. And based on that, we can say something about the shapes, like whether it is better to have a monotonic or non-monotonic single peak or multi-peak. This is going pretty heuristically, but still, I think it gives some insight that maybe we could go beyond this idea of Fisher information and look at the shapes. But there is something else that we did not consider, and that is usually ignored in tuning curve-based decoding of stimuli, and that is the issue of time. We assume that neurons, let's say a neuron has a tuning, and at its peak preferred orientation, it spikes at 20 hertz. So we assume that that 20 hertz is available arbitrarily fast. But obviously, neurons have to have some time. They need to accumulate certain number of spikes to get to a reasonable estimate of 20 hertz. So this arbitrarily fast responses that are not possible in actual systems. So we were then asking this question about not only the distortions, weak or catastrophic, but also the time it takes. So if we formulate this problem for time, then hopefully we can get some other insight about these tuning curves as opposed to just resolution and so on. So we start with a system where we have periodic tuning, because initially we formulated this question for single peak versus periodic tuning, place cells versus grid cells, or vision versus entorhinal cortex. So we have single peak tuning curves, and we can have multi-peak by changing the lambda of that. Now, if we assume that we have periodicity in the tuning, then obviously we can only represent things in a non-ambiguous way over certain region, like that is the wavelength of the tuning. So automatically we need, like if you want to work with periodic tuning, you need many, many modules, or you need a wide range of frequencies. We went for modules because it turns out that in entorhinal cortex, there are modules. Neurons have distinct frequencies as you go from, I don't remember which axis, but there are supposedly different modules of spatial frequencies. So that's the formulation of tuning curves then. So we have the amplitude, we can control the width, we can control the wavelength or the periodicity of it, and there is some baseline. When we have different, and of course it can be multidimensional, so it could be just one dimension like orientation, but it could be like orientation versus color, for instance. And then we have some modules in it, L modules in total, and for each module we take the harmonics. So the next one will be harmonic, like pictorially here shown. So in this case, now if we ask a question like, given this kind of tuning, what kind of activity pattern you will have? Well, it will depend on the time. So we are assuming Poisson spike here. So this is what will give you the activity pattern R, given a stimulus and given the amount of time. And of course, it will have some mean value, which is then how many spikes you will see in a given time. So with this now, we can calculate Fisher information. I won't bother you with the derivation, but you can see it here. And that derivation is, yeah, once you look at it, then this is where the periodicity enters in that. And this, of course, says that more periodic the tuning curves are, or higher the frequency, higher is the Fisher information. And this is then the old result of Lafayette, that, yeah, periodic tuning curves are better, they give you higher resolution and lesser errors. But that only works with these weak distortions. But then we were looking into something else in this. We said that, okay, let's look at not just the Fisher information, because Fisher information is a local measure, right? That's what I meant when I say that this applies only to the weak distortion. But when we are thinking of this threshold distortion, Fisher information has no clue about this. And to illustrate this, what Movitz did is that he gave himself some time to collect spikes to decode and measure the root mean square error. And at some point, the root mean square error approaches the Kramer bound, which is these dotted lines. Blue line is for single peak tuning, and red is for a periodic tuning. So they reach at some point, they hit the Kramer bound. Single peak tuning curves reach that earlier, multi-peak, they take some time. But now at this point, if you now look at so you do many trials, and every time you reach here, you look at the kind of error that you are seeing, and this is the error distribution. So error distribution looks pretty neat, and as Fisher information would suggest, that the variance is very small here, meaning periodic tuning curves are better than the single peak ones. However, this is the region. If you zoom in here, you find that there are actually errors happening, and more often those errors are in the red color, meaning they belong to the periodic tuning. So even though most of the time periodic tuning curves, they give you a very good estimate of the stimulus, they are prone to these threshold distortions. Now, one could say that these are very few errors, but I mean, there's a reason they are called catastrophic, because if you make this error, this has very serious consequences. So yeah, that's the issue that is happening now, that we have threshold distortions still continuing. And then we formulate this problem as a time problem, that how much time actually do you need to avoid threshold distortions? So this is how we pose this problem, that let's assume that we have two modules only, so single peak, and it has also a module which is periodic. Now, in this case, you get your joint likelihood given that you have one module which is single peak, another module which is multi-peak. Let's say the true stimulus is here, and this is your joint likelihood. Based on this, you will have some error. This is the true stimulus, so this much is the error. Depending on the frequencies in a periodic case, so here you are likely to get only one peak, right? Because you have one single peak, and then there are multi-peak ones. So you will get a single peak in the joint likelihood. Here, if you now look at the frequencies, so depending on what frequency you are choosing for your modules and how close they are to each other, you can actually end up with a very large error because of, let's say, beat formations. So you can have arbitrary large distances between the true stimulus and your joint likelihood. This kind of problem doesn't arise when you have just single peak tuning curves, right? So lambda is just one in this case. So with this heuristic, we say that, okay, we can't really analytically solve it, but we can try to estimate the threshold distortion. And we could say that, hey, given this delta, this error, how much delta can I afford? And obviously, the amount of delta that I can afford, it depends on my periodicity of the tunings, because eventually this is going to create a folded manifold, and I want to have my error smaller than the distance between the two folds of the encoding manifold. And that's what is shown here, that depending on the frequencies, and here we don't have two different frequencies, but multiple, so therefore this expression, so this is the maximum error I can afford. And now I can ask, like, what's the probability that my error is larger than this? And if that is the case, then we have a probability of a threshold distortion. So this is how we formulate this problem, and then we can actually calculate it. So time to avoid threshold distortion is, of course, dependent on the individual Fisher informations and the maximum allowed distortion. So smaller is this, larger time you need. So we can get some bound on amount of time you need to avoid threshold distortion with some probability. So if we calculate this, so the solid line here is this equation here. For single peak and for multi-peak, what we are changing is the scaling factor for different modules. So you go from here to this side, it becomes more and more periodic then. And obviously, as you become periodic, you need longer and longer time to get to the to avoid threshold distortion. And obviously, this is improving Fisher information anyway. So as you give more and more time, so that's fine, which is here. For periodic case, you get these harmonics, and they are just because, at some point they become subharmonics. So one module frequency is subharmonic of the other, and so therefore you get these large peaks. Otherwise, this is quite nice. It matches with our simulations. And so what it tells us is that in general, time to avoid threshold distortion is much larger for periodic tuning as opposed to single peak. And now one can ask, so this is what reviewers forced us to do, is to actually plot the distribution of errors. So here what you see is the root mean square error. These are the solid lines. And these dots here, they tell us what's the largest error. So in terms of percentile. And this is the amount of error that you are actually making, this absolute value of that. So red dots belong to periodic tuning, and at short time intervals, you are making very large errors. As you give more and more time, the blue dots, they drop down. So meaning the probability of seeing a threshold distortion in single peak tuning curve at 50 milliseconds is very, very small, and at 100 is basically negligible. Only weak distortion is seen, if at all. But periodic tuning curves, they keep showing us these errors. So that's what we mean by the problem, or this is what I think is the problem with periodic tuning, that even if you give them long time, there is always some chance that there will be a large error. Then we scale this up, and we go to more modules, in this case for 5, and we can again do the same calculations. And the result is the same, that as you make your tuning curves more and more periodic, you tend to need more time to avoid threshold distortion. These lower lines here, they are blue and red. They are for one-dimensional stimulus, and the orange and the green are for two-dimensional stimulus. Now, this is the Fisher information and the decoding time. So more decoding time, of course, Fisher information is higher. So that's, I think, the story then, that the periodicity of tuning curves, it imposes a limitation on these large errors. And you can avoid them if you give yourself more time. And this is yet another way to show this, that how this error distribution changes as a function of decoding time. So this is the time when you want to decode, and this is how the error is distributed. As you give yourself more and more time, then this error drops down. So that's basically the story. But then we try to put some bells and whistles on it. And one of the things that we were asked to do was to show it for a moving stimuli, because earlier we were just assuming one static stimuli after another. So here is one case where there's a sudden transition from one stimulus condition to another. And there also you can see that, of course, there are these weak distortions all the time happening. This is for single peak, and this is for multi-peak or periodic. And here again, so this seems smaller, but every now and then you see these errors. And this is something that we see all the time, even when the stimulus is actually moving as a function of time. So this is, let's say, temporal evolution of the stimulus orientation. This is for single peak. This is for multi-peak. So as a function of time, you keep making errors. So in the previous discussions, up to this point, what we were using was a maximum likelihood decoder. But here we change that, and we make it a little bit more neural, in the sense that there are these neurons which are which compete with each other. So there is this recurrent inhibition which selects the neurons which are high firing. So even with a more neural type decoder, you get exactly the same result, or qualitatively the same result as we had for the maximum likelihood decoder. Yeah, this is another way to show these error distributions. So upshot is that these are bad, meaning large distortion is very likely for multi-peak tuning curves. Other thing that is very bad for periodic tuning curves is, again, within this framework of avoiding threshold distortion, is the background activity. So this is the scenario where there is background activity. One is 0, one is 2 Hertz. And here it has an effect, but not so much. But if you look into periodic tuning curves, and for higher dimensions in particular, then this becomes really bad. So more higher the dimension, higher the background activity, longer time you need to avoid threshold distortion. And population size, of course, can correct for it. You can recruit a larger population to avoid this, but qualitatively, I think the effect still remains the same, that if you compare single peak versus periodic, you will always do better with single peak in terms of tuning, in terms of avoiding threshold distortions. I mean, the results are pretty systematically as expected, because as you increase the number of neurons, the minimum decoding time decreases. As you increase the background activity, it increases. But relative differences are quite stark. So this was all calculations. Is there any suggestion in the experiments for this idea? We found only one of this kind. In Freiburg, Marlene Bartosch's lab, they recorded boutons of entorhinal cortex cells in CA3 or CA1 or dentate gyrus, as well as the local cells. So these are the curves or data for local cells, and these are the entorhinal boutons, meaning grid cells, and these are largely place cells. And what they ask is that the color is the accuracy, how good it is. This is how much time you need. This is number of boutons or number of cells you need. So if we compare these two columns for the same accuracy, actually you are doing much better with neurons than with boutons. There are tons of caveats with this comparison here, because boutons and this is calcium imaging. There are all kind of other issues, but at least there's a suggestion in the right direction that periodic tuning needs more neurons, more time to get to the same accuracy. So all this was just a comparison of single peak versus periodic. But as I earlier said, that tuning can be of many different varieties. So you can have monotonic tuning. You can have a single peak, but each peak is very heterogeneous compared to each other. Or you can have homogeneous single peak ones. You can have periodic with different modules. You can have aperiodic and heterogeneous, and aperiodic and homogeneous. So all these possibilities then we can consider. And we actually took some data from this paper from Nakrum's lab, where they had given some idea about how the field size is distributed and how many multiple fields each cell has and so on. So we use this data to construct now different types of tunings, monotonic to aperiodic and homogeneous, heterogeneous, and so on. So in each case, you can look at how the firing would look like and how is the Fisher information. But as I said, Fisher information is not really a good measure when we are looking into large errors. So we go back to our root mean square error and how much time you need to avoid threshold distortion. Now, color code is a little bit messed up, but we could always look into single peak homogeneous, which is the blue lines here. And green would be, this beige would be periodic, and so on. So even with this, like if we consider aperiodic tuning, green and the orange ones, in each of these cases, whether you look at root mean square error, whether you look at the 99th percentile error or maximal error, in each case, turns out that at short times, single peak tuning curves win. In some cases, monotonic wins. But in most cases, there is a slight advantage over single peak ones. Periodic ones are particularly bad, but they are good over large time intervals. So if you have long time, then of course you will do much better with periodic tuning. And same with aperiodic, that also doesn't do so bad. So aperiodic or periodic, meaning non-monotonic tuning, these beige, green, and orange, in long time, they always do better than the single peak and monotonic one. But in short time, single peak, monotonic, they outperform. Yeah, that's then the field size distribution and so on. So this is just the details of this. Basically, I can close with this cartoon that Movitz made, that to us it looks like that if we follow this idea of representing information using tuning curves, then single peak tuning curves that we are more used to seeing in early visual processing, auditory processing, etc., at short intervals, they give us a reasonable accuracy. At large intervals, if you have long time, the accuracy doesn't improve that much. But good thing is that you don't make huge errors that you go outside the dartboard. Periodic tuning curves, they are very good in large. You get like really bullseye in long time, but in short time can be as bad as going outside the dartboard. So I think that's the way one can think of how speed accuracy trade-offs are perhaps inbuilt already in the sensory representation. In many ways, I think it makes sense that vision is fast. So you want something where you can get a reasonable estimate with good speed. Because if you miss it, it will have, I mean, you don't have so much time in natural world to look at things, and as opposed to variables like places, which I think are slowly moving, there you have more time and you can be more accurate. But this also makes us think that, I mean, why bother about such high accuracy for places? Perhaps they are representing some other thing. Maybe we are interpreting it as a place, but maybe brains are representing something else with entorhinal cortex grid cells. So I am a cricket player, and I often ask myself like where it is useful. And I think there's one place where vision has to be extremely good while playing cricket. The player has to observe the seam and literally the angle. That angle predicts how this ball will swing. And if you miss that, that's really catastrophic. And vision, I think, is super important there. And I think it's reasonable to think that single peak tuning is much better a choice to work with than grid cells. Yeah, so thank you so much. That's all that I wanted to say.
Thank you, Arvind. Very interesting. So Arvind, I'm still trying to figure out how one defines an ensemble. So when you say it's 50 cells or 100 cells, so are we just saying the 50 or the 100 cells just physically closest to a given cell, or is there something more to that? Because if you look at the primary visual cortex, the cells that are most connected to a given neuron are not necessarily the ones that are in the immediate vicinity. Those are the cells that are most excitatorily connected are ones that are a little further away, connected by horizontal connections.
So in this case, we are not really making any assumption about local connectivity. What we are thinking is that local connectivity gives rise to these shapes. Right? So you have this tuning. So you need recurrent connectivity of E to E and E to I and I to E to get to these shapes. Once these shapes are there, then let's say V1 is the representation is V1 are being interpreted by V2, for instance, or V4. They can sample from anywhere. So there, the space notion is not there, like whether they are connected or not, because we kind of, I mean, it's a very good question. Like, what's the role of recurrent connectivity in this kind of coding? For us, we took a simpler route and we said that, well, recurrent connectivity only give rise to these shapes.
I see.
It's inherently taken care of by the shape of the tuning curve.
Like pretty much like following the notion of, let's say, Heim's work on tuning curves in the visual system, or Yasser Rudy's work on entorhinal cortex grid cells. So they always use recurrent connectivity to generate these shapes. So that's what we are also believing. And they create correlations, of course. And then this whole discussion, right? That given some correlation background activity, what kind of tuning would be good?
Hmm.
So that's how the thinking gets looped through.
And just one other small question. So with the periodic tuning, is it the case empirically that neighboring cells have simply shifts of, so phase shifts, or do they also have shifts in the frequency?
So neighboring cells only have phase shift.
I-
And in entorhinal cortex, it turns out that there is one axis along which if you go, then the frequencies change. So within one plane, as far as I know, all the cells have same frequency but different phases.
I see, I see. And if you were to pool cells with different frequencies, how different do the results look? Instead of pooling simply cells with different phases.
Yeah, so this is what we try to do here with periodic and we don't have that, periodic inhomogeneous.
Hmm.
Okay, I should have that. So yeah, basically the results look the same when we have periodic and inhomogeneous.
I see,
Yeah.
I see. Hmm.
Yeah, here we basically went through the harmonics of it, but yeah, we should have that as well in this extra column.
Arvind, may I ask a rude question?
Yes.
Usually when someone skips over a slide, there's a good reason for it.
Yeah.
And when you were talking about moving stimuli, I noticed that you jumped over. You had a slide that showed basically stepwise stimuli and slowly changing stimuli. And I was wondering, because I'm really interested, and not just in everything that you said, but particularly in this comparison contrast. I wonder if you could say something about what's on this slide.
Okay. So what we do is that we say that there is a stimulus here and there's a step-like change.
Yeah.
So you have a stimulus with, I don't know, 0.25 orientation or whatever, and here it is, 0.75. So there's a sudden change, and what your neurons now have to decode is like, what's the value of the stimulus, right? That's the task. Blue line is the true stimulus, and these red ticks are the estimate from the population. And the way the decoding here is done is, so we have these inputs coming in, neurons are activated, and then there is this inhibitory circuit, which does the winner-take-all kind of computation here so that we get sparse representation. And from there, we are then reading out which neurons are activated. And based on that, we get some notion of an error. So here, this is for single peak tuning population. So here the stimulus one ends and it changes the state. So what you get is a small error, of course, in the beginning, and then very quickly the error converges to the next new value. If you look into the periodic case, you have the step-like change. So here your neurons are still decoding the first stimulus, then they go to the next one. But even when they are in the next one, there are these errors that are happening, and even for the first stimulus. So what we are highlighting here is these extra ticks which are far away from the blue lines, those are equivalent to the threshold distortion. That's what I wanted to indicate.
Okay. Wow.
So if the stimulus is fast changing or fast moving, then periodic tuning curve will make larger errors than the single peak ones. That's the claim. So maybe as an experiment, perhaps if, I don't know if how fast we can change places, and place is also a bit difficult because eventually vision and other senses are also involved. So it's very difficult to construct an experiment. But if we could do one, then I would say that maybe like if you compare place cells versus grid cells, you would decode better at shorter time intervals with place cells than grid cells. And that's what this data suggested as well.
I'm going to have to think about whether, without going to places, just sticking within the visual domain or the auditory domain, one can construct stimuli that mimic those kinds of different kinds of changes. So I can imagine you have a second order derivative of or a second derivative of some stimulus, which could give you real challenge for this system.
Mhm. Yeah, I'm very curious to find out like what kind of stimulus we can design and test this.
Yeah, yeah, yeah.
Cool.
I think, Arvind, in principle, you could accomplish that without going to places.
So I'm happy to hear that.
Yeah.
This just shows my ignorance of the experimental literature as well as the capability of experimentalists.
Well, this is this conversation between experimentalists and computationalists is an important conversation, in part because we on the experimental side often need the inspiration of thinking outside our boxes,
Yeah.
and imagining the kinds of questions that are raised by models like the one you presented.
Yeah, so I'm very keen to hear like if you can design something and
Yeah.
test this, I guess would be great. Yeah.
If anybody can, Bob can.
Can I ask one more a quick question? This is a technical question. Could you say something about information limited correlation?
So in simple terms,
Yes, please.
so the way they formulate it was that, so physically speaking, so the notion of information limiting correlations is that imagine that not only that like why is this dot here, right? This estimate here. This could be because that you added some, no, it could mean, how do I say, yeah. So there are two sources of noise. One source could be that this response of the neuron can change by itself, right? So you get,
Yeah.
for the peak, you get some value here. But the other source of noise could be that as if the tuning curves themselves are shifting, right? So imagine
Yes.
that two neurons, their tuning shifts a little bit. They are never perfectly tuned to, let's say, 90-degree. So in one trial, they are a little bit more tuned to 85. And so if you imagine that tuning curves are shifting, then the kind of correlations that will arise, those amount to information limiting correlations. Geometrically, what it means is that these correlations, they get, where is that slide? Geometrically, it means that these correlations, they get aligned to the encoding surface. And if they get aligned to the encoding surface, there's no way to separate them.
Okay.
So that's more technical way to say
Yes,
this. And
that's fine.
physically is the lilting of the tuning itself.
Good. Thank you. Thanks.
So that's where this overlap comes, right? So we are saying there's a sensor noise. And what Ruben would say that, no, it's not sensor noise. Maybe it is actually tuning which is shifting.
Mm-hmm.
Right? And to us, it looks like much more, it's likely that there's also sensor noise, right? Who says that, sensors are perfect? So,
Arvind, it's nearly midnight for you, right?
almost.
Oh,
Yeah, we should let you get to bed. Thank you so much for staying up this late and sharing this wonderful work with us.
If you've got more questions, you can share with me later after the meeting, and I can email Arvind those questions.
okay.
Great, thank you so much. And yeah, I apologize for being late as well. So unintended, but yeah, I thank you for waiting as well.
Thank you for a really wonderful talk. Thank you.
Thank you.
Thank you.
So much. Bye-bye.
Thank you.
Have a good night.
Good night.
Bye.
I can try to join this one and we prefer.
How does the original channel noise?
Sure, yeah.
What do you mean by that?
So Bob and
Two
Gadi,
types of noise, he says.
Um,
One
Mikhail
depends on the
is inviting a person who works with children with disabilities, who places children with disabilities in the school system, in the Boston public school
Oh.
system.
Or the Cambridge, I can't remember, one of them.
I see.
Cambridge or Boston.
So you're very welcome to stay on, but she will
No,
likely.
I don't think so, but I'm looking forward to the next meeting. Peter is going to, yeah, right. It's going to be a really good one.
Yes, yes, it would be lovely to
And-
have you join Bob.
Yeah, good.
Uh,
Good,
Yes,
Good.
thank
Okay.
you for joining.
Okay.
us.
Yeah.
Okay. And thank you for inviting me. Okay,
Always a
All right,
pleasure.
anyway.
bye-bye. Bye.
Bye, Bob.
I,
Yes, so.
Yeah.
So, so Gadi, I was saying that we'll have this conversation, a brief conversation with Megan Singh, to talk about the work that she does and see whether there are potential connections